Hello everybody, this is Dr. Christopher White, and today we're moving on to lecture number 15, where we'll be dealing with Paleozoic life history. It's going to be split in two parts. So the first part of this presentation is going to focus on the invertebrates, and the second half of the presentation is going to focus more on the vertebrates. Alright, so let's get going. Okay, so the start of the Paleozoic is marked by the sudden appearance of large quantities of life with shells. Now this is very different to the rocks that came before them, in the Proterozoic, because as we've already discussed, life in the Proterozoic was either single-celled, and therefore didn't fossilize very well, soft-bodied, didn't fossilize very well, or, when we did have macroorganisms that had shells, well, those shells once again were made typically of organic material, and so once again, they didn't fossilize very well. So what we have in the Proterozoic and in the Precambrian as a whole is a distinct lack of fossils. Now, when we look at the rock record, all of a sudden we see that as we go from the Precambrian into the Cambrian, we suddenly get this massive increase in the amount of diversity that we see. We also see a lot of the organisms are macroorganisms, so we can actually see the fossils, and they had hard shells, so they fossilized relatively well. And so this essentially was used to define the boundary between the Cambrian and the Precambrian, this sudden appearance of large quantities of shelly life. So the early Cambrian is marked by a rapid increase in fauna, particularly those with skeletons, and this is referred to as the Cambrian Explosion. So the increase was problematic for those 19th century scientists who supported evolution. And of course, evolution hypothesized that essentially it was a gradual process. So obviously before the Cambrian explosion, you would have to have a build-up of several generations of organisms that led you to the Cambrian explosion. Now, when we look at the rock record, because we didn't have many macrofossils, remember the Ediacaran fauna weren't discovered until relatively recently. So essentially, when they looked at Precambrian rocks, there were none of these macrofossils. Then all of a sudden, in the Precambrian, explosion, life everywhere. And so this was problematic because, if you're saying, well, it was obviously due to evolution, well, what you needed was a gradual build-up. You can't just suddenly have life exploding out of nowhere. That doesn't work for evolution. Now we know that we've gotten around this problem because now we have fossils of these soft-bodied organisms, steadily becoming more and more evolved throughout the Proterozoic, and we also, of course, have the appearance of the Ediacaran fauna right at the end of the Proterozoic that shows us that these organisms have reached a point of a very high degree of complexity before we get this jump into the Precambrian and we get the appearance of all this life that has shells. So we should remember that the explosion was fast by geologic standards. Okay, it occurred over a period of 20 million years. So to you and me, that's actually quite a long period of time. From a geologic point of view, it was pretty rapid. So when we use the word explosion, we are talking about in a geologic sense, not in the kind of sense that we would appreciate on a human timescale. So the majority of the Ediacaran fauna died out at the end of the Proterozoic. However, why so many animals suddenly appeared at the start of the Cambrian is hotly debated. So, when it really comes down to it, the fauna of the Ediacaran, the Ediacaran fauna, should I say, probably died out because they were just outcompeted. So, conditions between the end of the Proterozoic and the start of the Cambrian aren't too different, and so, the reasons for the Ediacaran fauna dying out were probably just due to them being outcompeted by the new organisms that made their appearance. So anyway, why do we suddenly get this increase at the start of the Cambrian? Well, there are actually four main possible reasons to explain this. The first one is climatic. So the Cambrian explosion, the Cambrian environment was warmer than the previous Proterozoic era. So you have to remember, during the Proterozoic, there were, periods of widespread glaciation. It would lead to, on the whole, lower global temperatures. And so this means that for the vast majority of the Proterozoic, chances are ocean water temperatures were lower. And, that would have made it a bit more difficult for life to get established. Life prefers, moderate to warm conditions. And so as we move from the late Proterozoic into the early Cambrian, we begin to see this increase in global temperatures, and that's going to have affected global water temperature. So we would have seen global water temperature steadily going up as we move from the late Proterozoic into the Cambrian. So the climate would have been a contributing factor to some degree. We also see changes in ocean chemistry. So important factors when it comes to changes in ocean chemistry include concentrations of calcium dissolved in seawater and, of course, pH. How acid or alkaline is the seawater? So concentrations of calcium could have reached the saturation, could have reached supersaturated to saturated conditions, that exist. Do you know what? I've got that sentence completely wrong. Let's go back to the beginning of that one. So concentrations of calcium could have reached the saturated to supersaturated concentrations we have now. So the vast majority of water that we deal with when it comes to the oceans and when it comes to the water on land has a very, very high concentration of dissolved calcium in it, typically in the form of dissolved calcium carbonate. And throughout the Proterozoic, throughout the Precambrian, should I say, these concentrations were building up and up and up and up and up. And by the time we make it into the Cambrian, it's probable that the concentration of calcium carbonate in seawater was near saturation levels. So, and those are the kind of conditions we have now, by the way. So the buildup of calcium carbonate in seawater would have made it easier for organisms to make shells out of calcium carbonate, because there would have been large quantities of calcium carbonate dissolved in seawater that they could have used to make shells. And so, it could just be simply down to the fact that life was waiting for calcium carbonate to become, generally available, at which point life could, start to use it on a large scale to make lots and lots of shells. So the increased concentrations of calcium carbonate would, calcium dissolved in seawater, would not only have made calcium carbonate more common, it would also have made calcium phosphate more stable as well. And so calcium phosphate is another material that is used by some organisms to make shells. In fact, calcium phosphate is the stuff that our bones are made out of. So the increase in calcium carbonate would also have aided in the formation of in the evolution of organisms that like to use calcium phosphate to make shells over calcium carbonate. Another reason that the increase in calcium carbonate concentrations would have been helpful is that it would have helped to have neutralized essentially any acidic compounds in the oceans, and this would have helped to take the water to essentially a near neutral pH, which once again is what life seems to prefer. On the whole, life likes conditions to be warm to at least comfortable, and typically life likes the pH to be somewhere around neutral. So as we move into the Cambrian, we also see an increase in the amount of continental shelf environments. So if you remember, right at the end of the Proterozoic, we have the supercontinent Pangaea. And as we move from the Proterozoic into the Cambrian, Pangaea is falling to pieces. And so what does this mean? Well, it means that Pangaea is all of a sudden breaking up, producing lots of smaller continents, and of course all of those smaller continents have coastlines, and so that means the amount of essentially passive continental margins is on the rise. Because, think about it, a single supercontinent only has one coastline, and compared to the total volume of continental crust, within that supercontinent, the coastline will not have actually been that big. Now, when you take all that continental crust and break it into lots of smaller pieces, all of a sudden the amount of coastline available shoots up. And so this means that there would have been lots of passive continental margins at the start of the Cambrian, and life loves passive continental margins. Life likes to form in essentially nice, boring environments where not much is going on. And so a passive continental margin is a great place for that to happen. And also, when you're talking about life on the oceans, well, life in the oceans tends to like to form in regions of the ocean where there is available sunlight, and so that the best environment for that would be typically in areas that have water depth between about 0 and 200 meters. So once again, all of these new passive continental margins that make an appearance are going to encourage the formation of life. And then as we move into the Cambrian, we also see the first appearance of organisms that are definitely predators. And so all of a sudden, we have a situation where we have an arms race starting. And so if you're an organism that is has suddenly become a prey item, well, naturally, if you can evolve a shell, you're going to be at an advantage. Okay, so the evolution of predators, the first appearance of definite predators, means that all of a sudden having a shell is helpful. It offers you some protection. So which one of these factors is more important is uncertain. It's highly probable that the Cambrian explosion is just a reflection of all four of these factors coming about at approximately the same time right at the start of the Cambrian. So before we actually get the Cambrian explosion itself, we do actually see the emergence of a group of organisms which are called the small shelly fauna. So if we remember, we actually see the first traces of shells and mineralized coatings appearing with the Ediacaran fauna. So if you remember, there was that organism, Kimberella, I think it was. And Kimberella it looked like a limpet almost. Essentially, it had this hardened, kind of conical shell, and the organism itself would obviously, gather itself under the shell for protection, if it had to, do so. And so from what we can probably guess, the shell of Kimberella was probably made of chitin, which is the same material that insect shells are made out of. So it was organic. Now, if you also remember when I was talking about the Ediacaran fauna, there are also some indications of shell fragments, and these shell fragments were made of material like calcium carbonate. Now, these shell fragments are very, very small, and typically, we don't know what organism they belong to. But the fragments of shell that occur around the time of the Ediacaran fauna does suggest that there were some organisms that had the start of what we would classify as a proper shell, or maybe just a mineralized coating of some kind. So we know there were we had the start of shells making their appearance with the Ediacaran fauna right at the end of the Precambrian. Now, these were followed by the small shelly fauna, and if you look at the picture behind, you can see some members of the small shelly fauna. And these are typically sub-millimeter to several millimeter long organisms, or parts of organisms. So quite a few of these fragments that we see in the small shelly fauna are often pieces of shells belonging to larger organisms. So they're bits of shell belonging, or mineralized coatings belonging to organisms like sponges, brachiopods, or echinoderms. Echinoderms are the groups that include sea urchins and starfish. So the small shelly fauna are made actually of a range of materials. So some of them have their shells made of silica, some of them have shells made of calcium phosphate, and some of them have shells made of calcium carbonate. Now, the interesting thing is that all forms will occur in the same environment at the same time. So that's an interesting feature. So typically in a modern environment, there will often be something about the environment that will encourage the formation of a certain type of shell. Some environments would encourage the formation of a silica-rich shell. Some environments would encourage the formation of a calcium carbonate or a calcium phosphate shell. Now, the fact that we don't see this distinction in the early Cambrian suggests that the concentration of calcium or calcium carbonate dissolved in seawater were not the only controlling factor when it came to their evolution. So if you remember on the previous slide here, I was talking about how the changes in ocean chemistry could have been a major factor in the formation, it could have been a major causation behind the Cambrian explosion. Now, when we look at the small shelly fauna, what we're noticing is that the concentration of calcium or calcium carbonate does not appear to be the only controlling factor in their appearance. If it was, if it was all to do with the amount of calcium carbonate dissolved in seawater, and we saw that, as we move into the Cambrian, the amount of calcium carbonate has gone up significantly, well, that's going to encourage the formation of organisms that have calcium carbonate shells. And so we should see the shelly fauna being dominated by lots of calcium carbonate shelled organisms. If it was all due to the amount of calcium dissolved in seawater, well, we should see that the small shelly fauna would be dominated by calcium phosphate and calcium carbonate organisms. but we don't see that. We see in the same environment, we see silica shelled organisms, calcium phosphate shelled organisms, and calcium carbonate shelled organisms at the same time. And so this shows us that when it comes to the the evolution of life in the early Cambrian, the concentration of calcium or calcium carbonate dissolved in seawater was not the only factor controlling life's appearance at that time. Now, most of the small shelly fauna that we see are fragments of larger organisms, with only a few small members of the small shelly fauna actually representing discrete organisms. And so when we look at some pieces of the small shelly fauna, so let's look at number 12 here. That looks like the kind of plate you might get from an echinoderm, from a sea urchin. So chances are that is actually a fragment of a larger shell. Then you compare organisms like 1 and 5, you can actually envisage that they may have been discrete organisms in their own right. The organism itself may be lived up towards the top of the end of the shell here, and these may well have been earlier portions of the shell as the organism grew and got larger, more segments were added, and you can see the same thing here. So some of these organisms, some of these fragments should I say, would have been from larger organisms, some of them would have been from smaller organisms, or discrete organisms should I say. But nevertheless, it does show us that we have organisms with shells now developing in the very early Cambrian. These organisms were very small, but they will evolve into much larger shelled organisms. So the small shelly fauna were followed by the appearance of large skeletalized animals. Now you need to bear in mind that the term skeletalized doesn't always mean skeleton in the sense of our skeleton. So we have an internal skeleton. It also refers to animals that have exoskeletons, so animals that have their skeletons on the outside in the form of a hard shell or carapace of some kind. So one of the questions is, well, when we move into the Cambrian, we see a lot of organisms that have exoskeletons. They have shells or carapaces on their outside made of calcium carbonate, calcium phosphate, or silica. And so why? Why do these organisms favour having an exoskeleton over having an internal skeleton? So that's the next question we need to think about. So the first one is that it provides protection from ultraviolet rays. So once again, when we go back to the Proterozoic, by the time we go from the Proterozoic into the Cambrian, the oxygen concentration in the atmosphere is probably only about 10% of the current amount. So at present we have about 21% oxygen in our atmosphere. At the Proterozoic Cambrian boundary, that concentration would probably have been somewhere around 2 to 3% of the atmosphere. So as we have less oxygen in the atmosphere, that means we have less capacity to produce ozone. Obviously, as I'm sure you know, the ozone layer is one of the things in the Earth's atmosphere that helps to offer a significant protection from ultraviolet radiation coming from the sun. So a non-existent or a much weaker, thinner ozone layer at the start of the Cambrian would have allowed significantly higher concentrations of ultraviolet radiation to make it to the surface of the Earth. Now, as I'm sure you know, ultraviolet radiation is damaging to organic material, and so having some kind of protection from ultraviolet radiation would have been extremely helpful. It would have, significantly reduced the amount of damage that, animals were taking from these ultraviolet rays. Number 2, it helps prevent drying out in an intertidal environment. So what we see obviously life begins to make its first appearance in the oceans, and we very quickly begin to see that life shifts to not only living in a fully marine environment, but also into the transitional environment, the environment essentially where sediments are exposed to the atmosphere for portions of the day, and for portions the other rest of the day that area is underwater. So having an internal skeleton like ours is not an advantage in that situation. So if you're an organism that prefers to live in water, and you have an internal skeleton like ours, well, you have relatively little protection during the periods of the day when there is no seawater covering over the sediments that you're in. In that situation, you're going to start drying out, and that's obviously going to lead to death. So having an exoskeleton, think of an animal like a clam or an oyster, all of a sudden becomes extremely advantageous. So it means that when the sea goes out and you're left high and dry on the sandbank, well, all of a sudden, all you have to do is close your shell up, close it really, really tight, and that means essentially you can trap, a small quantity of water inside your shell. That's going to help, keep the organism inside moist, and so that's going to avoid drying out. And so having an exoskeleton allows you to avoid drying out, and therefore it allows you to move into these, transitional environments, beaches, lagoons, deltas, so that is quite an important evolutionary step. It also allows animals to increase in size and provides attachment points for muscles. So when it really comes down to it, an exoskeleton is a lot easier to put together than an internal skeleton. So once again, just think of our skeleton. Think of the sheer number of bones that are in it. Think of think of all the muscles and the tendons that are required to keep our skeleton from essentially falling to pieces. If we didn't have the muscles, didn't have the tendons, we would just become a flat pancake on the ground. So then think of something like an oyster shell. An oyster shell has two halves. It's very, very simple. Now also think of an oyster shell. If you've ever tried to break an oyster shell, ever tried to snap it, you will know that an oyster shell is an extraordinarily robust piece of kit. Once again, compare that to our skeleton. There's a reasonable chance that, when you were young, you fell out of something and you broke your arm or your leg. Our skeleton is on the whole relatively fragile. So exoskeletons tend to be far more robust and far simpler. Now, because they're relatively simple, that means the organism can, increase in size with minimum fuss. It doesn't mean there's significant redesigns required if an organism begins to evolve to become larger and larger and larger. Okay? It also provides attachment points for muscles. So when you think of, an oyster shell, a nice big heavy shell gives you the capacity to attach big, strong muscles to it. Okay, so imagine a situation where you had an organism like an oyster, and it only had a very, very thin shell, but it had big, powerful muscles. So when it tried to close up the shell, if it had big, powerful muscles, it would pull the two halves of the shell together very, very tightly. And if that shell was very, very thin, well, obviously that shell would then break because the muscles would be so powerful. Now, think of an oyster shell, a modern oyster shell. It's very, very heavy, very, very chunky, and so when when those big, powerful muscles pull the two halves of the shell together, well, the shell can take the strain of those big, powerful muscles and the forces they exert on the shell. And so having a simple, robust exoskeleton allows you to essentially attach very, very large, powerful muscles to it. And that's also advantageous, especially when you are dealing with some kind of environment where you're in an intertidal environment, when you really have to keep the parts of your shell closed up to stop you drying out. And this also allows for extra protection from predators. So if you're an organism like an oyster, well, one of the ways that the predator can get into the shell to try and eat you is by simply pulling the two halves of the shell apart. Now, when you think about oysters, they have these big, powerful shells, big, powerful muscles. That makes it very difficult for organisms to pull the shell open to get to the oyster inside. And so, once again, having these big, strong muscles attached to a big, thick shell is advantageous from, in the case of in the case of not being eaten, essentially. And obviously, having an exoskeleton provides, significant, protection from predators compared to an internal skeleton. So, just think of the situation. A predator goes after something like an oyster. It has to get through the shell to get to the soft stuff inside that it's actually trying to get to eat. In contrast, when it comes to us, well, a lion sees us walking across the Serengeti. Essentially, we have zero protection from that lion. So having an exoskeleton obviously provides significantly greater protection from predators compared to having an internal skeleton. So, and we can actually see we have fossils in the fossil record that show damage from predation, predators trying to get into the shell, but we can also see that from the fossil that the organism actually survived that attack, and so that we have clear evidence that shells work. Okay, so we've been going for over 20 minutes. This is a good place to take a break. So pause the presentation for a minute, get up, have a walk around, go and get a glass of water, and come back in 3 or 4 minutes, please. Okay, so we've dealt with essentially the changes that could have encouraged the formation of shells, and we've dealt with why having an exoskeleton is an advantage. So the final thing to think about is why marine organisms actually develop shells. So the first reason is it's a good way of getting rid of excess calcium. So if you're an organism and you're either eating large quantities of sediment or you're filtering large quantities of seawater through your body, well, obviously that means you are going to have a buildup of minerals dissolved in that seawater. So as we've already discussed, there's going to be calcium carbonate dissolved in that seawater, isn't there? And sometimes when the seawater containing that calcium carbonate comes in contact with organic tissue, it's going to encourage calcium carbonate to precipitate out of the water onto the organic tissue. Now, unfortunately, if that organic tissue, for instance, is your intestine, well, that's going to inhibit your ability to remove nutrients from whatever you're eating, and so eventually that could lead to you dying. So what you really want to do is you really want to have a method of getting rid of any excess calcium that builds up inside your body. And so simply sweating out that excess calcium onto your external surface is a good way of getting rid of it. And if you think about it, if you sweated out enough calcium carbonate over a period of time, eventually it's going to end up forming a very crude calcium carbonate carapace, isn't it? A very crude calcium carbonate exoskeleton. So to begin with, that's one of the ways, one of the reasons for developing a shell. It's simply a way of getting rid of excess material that you would have, ingested during, essentially the intake, by intaking seawater. So the next reason is that exoskeletons especially are biologically cheaper and easier to build than internal skeletons. Okay, plus they have much larger attachment points to which you can attach big powerful muscles. So yeah, exoskeletons are just easier than the type of skeleton that we have, these internal skeletons. So evolution is obviously going to favor, at least initially, the more straightforward approach. Okay, and so having an exoskeleton consisting of just a few segments, minimum of 2, is going to make your life a lot easier. So an exoskeleton is going to be the more straightforward thing, and so evolution is more likely to go that way. Obviously, as we've also discussed, the exoskeleton, so once again going back to the oyster shell, exoskeletons can be very heavy, very chunky, and so that's going to allow you to attach more powerful muscles to them, compared to something like our skeleton, which if you were to attach a muscle that was too powerful to it and you tried to move in the wrong way, essentially you could end up breaking a bone quite easily. So the advantages are quite clear in that respect. And then there is also the changing conditions. So the cold conditions of the Proterozoic meant that organisms had insufficient energy for biomineralization. So if you're going to have a shell, well, a shell is going to take up some energy to form. Now, if you're an organism and you're living in an environment that is relatively cold, you are going to be using as much of your energy as possible to make sure you stay alive. Having a shell will be a secondary concern to you. If you don't have enough energy to make a shell, you're not going to have one. It's going to be a waste of energy. So when we look at exoskeletons in cold water, in modern cold polar environments, what we see is that organisms in those environments tend to either not have shells, or if they do have shells, those shells tend to be rather thin. And so that's because they are focusing on using any energy they get, any energy they produce from ingesting materials to essentially keep themselves warm, make them grow, something like that. Essentially, having a shell is relatively low on their list of priorities. Now, what we see as we move into the Cambrian is we see global temperatures increasing, and therefore we see seawater temperatures increasing. And so this is going to give us a situation more like a modern tropical biota, faunal group of organisms. And so it's going to mean that all of a sudden having a thick exoskeleton, having a thick shell becomes, from an energy point of view, viable. So all of a sudden you're living in these warm water environments, there's going to be plenty of food around, and so this now means that you're in the right environment to essentially form one of those shells because you have the excess energy. And of course, having a shell also gives you an advantage when it comes to predators. So, of course, predators will also be present at at a higher abundance in these warm water environments because there's more life as a whole. So it's clear that once shells begin to appear, marine organisms with them will most definitely have an evolutionary advantage in the vast majority of cases, and so we will see them very, very quickly outcompeting the vast majority of organisms that do not have shells. So we're going to take a moment now and we're going to think about the modern marine ecosystem, and we're going to think about all the different parts of it. So by looking at modern marine organisms and the adaptations that they have developed in order to better succeed in their preferred ecosystem, we can gain an insight into ancient species. So to begin with, organisms are separated based on their preferred ecosystem. So organisms that live in the water column, so they're swimming around actually in the water, are termed pelagic. And those are, for pelagic organisms, are subdivided into two groups. There are the planktonic organisms, and those are organisms that are free-floating. So they're just floating around in the water. They don't really have their own capacity for propulsion. They move around wherever the currents take them. And these organisms are typically split into two broad groups. There is the phytoplankton. So those are free-floating organisms that essentially photosynthesize, so they're plants. And so that includes groups like diatoms, dinoflagellates, and algae. And then we have the zooplankton, and those are animals like jellyfish, foraminifera, radiolarians, that essentially are animals, i.e. they have to ingest nutrients in order to survive. They cannot produce their own food. So essentially, pelagic organisms, the first group is plankton, so they're free-floating, and there are two subgroups of plankton: phytoplankton, those that can produce their own food, and zooplankton, those that have to ingest their own food. Then we also have a group of organisms which we refer to as nekton or nektonic organisms, and those are the swimmers. And they're mostly vertebrates, so think of fish, sharks, etc. But there are some invertebrates, including the group that has cephalopods in it. Cephalopods are the group that includes squid, octopi, and cuttlefish. So these are organisms that have the capacity to propel themselves through the water. Now then, there's a group of organisms that obviously live on the sea floor, and they make up a group which is referred to as the benthos or benthotic organisms. So they are split into three subgroups. There are epifauna, so those are animals that live on the seafloor, so oysters, barnacles, etc. There's epiflora, those are plants that live on the seafloor, so think of kelp, essentially seaweed. And then we have infauna, and these are animals that live and move through the sediment. So think of organisms like worms, clams, any animal that kind of buries itself down in the sand or the mud on the seafloor. So these benthotic organisms, those benthotic organisms that stay in one place are also referred to as sessile, while those that move around are referred to as mobile. So think of an oyster. An oyster, once it forms an attachment to a surface, pretty much isn't going to move. It's going to stay pretty much where it is. So that would be a sessile organism. On the other hand, think of something like a sea urchin. So a sea urchin has the capacity to move around the seafloor, as it hunts around for food. So that's going to be a mobile organism. So as well as considering where they live and whether they move, we must also consider how they obtain their nutrients. So obviously, as you can see, there are 5 subgroups essentially of dealing with how these organisms that live in the oceans are getting their nutrients. So the first group are the photosynthesizers. That's essentially the group that has the capacity to use the process of photosynthesis to create food. And essentially that's the group that includes marine plants. So then there are suspension feeders. So suspension feeders consume microscopic animals and plants, and also some nutrients from seawater. And so what these organisms will do is, essentially they have the capacity to filter out, small microorganisms from, large volumes of water, and these microorganisms will then be, typically, passed to some kind of mouth or gut where they will be digested. These organisms will also have the capacity to take some nutrients from seawater directly in order to allow them to survive. So then we have a group of animals that we term herbivores. So those are the plant eaters. So those are organisms that will actively move around on the hunt for stuff to eat. And, well, should I say, for plants to eat. Now, then we have carnivores. Those are meat eaters. So they're actually going to go after organisms like the herbivores, the suspension feeders, the photosynthesizers. So they are typically meat eaters. They are out to eat other animals. And then we have the sediment deposit feeders. So those are organisms that ingest sediment and they will extract nutrients from it. So they'll eat sand, and as the sand passes through their gut, any small organic molecules, pieces of organic material within that sand will be digested, and the nutrients will be extracted. And then anything the organism cannot digest will simply be pooped out the other end. So as you can see, there are 5 distinct groups of organisms, based on how they feed. So you can see that, when we're discussing organisms in the marine environment, we're thinking, number 1, do they live in the water? Do they live on the seafloor or in the seafloor? Do they, are they essentially stuck in one place? Do they have the capacity to move around? And then we have to think about, how are they getting their food? Are they making it themselves? Are they filtering out small particles from the water? Are they eating plants? Are they eating other animals? Or are they simply ingesting large quantities of sediment and extracting, nutrients from that? And so this diagram is taken from the textbook, and you can see, it covers the range of different organisms and different environments. I'm not really going to go into it because we've got to keep moving. So, as you can see, there's lots of complexity, but don't worry, it gets more complicated. So an ecosystem will also include several what we refer to as trophic levels. And essentially those are tiers of food production and consumption within a feeding hierarchy. And it's actually easier than it sounds. So in the beginning, there are primary producers. Those are the autotrophs. So those are animals that make their own food. So organisms like phytoplankton. Okay, or simply, the photosynthesizers, plants, seaweed. So these primary producers will be eaten by primary consumers, which are of course the herbivores or the suspension feeders. So they're going to take these autotrophs and they're going to eat them, and then the primary consumers are going to be eaten by the secondary consumers, the carnivores. And obviously then the secondary consumers will then be eaten by tertiary consumers, and the tertiary consumers will then be eaten by the quaternary consumers, and so on. So you can see you have a system that contains several distinct levels. And obviously as you go up from one level to another, typically the number of animals in each level decreases as you go up until you end up with the apex predator, essentially the predator right at the top of the tree. And there are also transformers and decomposers. So these are bacteria that break down unconsumed organisms into inorganic compounds which can then be used by plants and suspension feeders. You know what? It should actually be bacteria and fungi, not just bacteria. And yeah, they will break down organic material. They'll essentially, break it down. They'll transform it. They'll break it down to its, component parts as part of their, part of their digestive process to extract energy to survive. And essentially, the waste products they produce can then be used by other organisms, particularly plants and suspension feeders, to survive. And you can see once again, here's another diagram from the textbook. You can see the very complex interrelationship between each of these different environments. We're not really going to go into it because if you wanted to spend some time looking at it, feel free. But I think we've covered reasonably well in the past couple of slides. Okay, so we've been going for 40 minutes now. So once again, pause the presentation, get up, have a walk around, get a glass of water, and come back in a few minutes, please. Okay, so now let's get back to the Paleozoic marine invertebrates. So if we try to deal with the changes that occurred in each of the invertebrate phylums during the Paleozoic, our minds would literally melt, because there's just so much going on, especially during the Cambrian. So instead what we're going to do is we're going to look at the communities as a whole and look at some of the broad changes that occurred. I'm going to keep it relatively simple and relatively unfocused, essentially. So we need to remember that the Paleozoic was a time of significant change. We had marine transgressions producing expansive, shallow, warm seas. If you remember, during the early Paleozoic, we have the Soak and the Tippecanoe, and during the late Paleozoic, we have the Kaskaskia and the Absaroka. In terms of the Soak, the Tippecanoe, and the Kaskaskia, they resulted in very nice, shallow, warm, clear seas, which would have been perfect for for life to live in. The Absaroka wouldn't be quite so good, but still it would have provided lots of environments in which life could establish itself. And we need to remember as well, we have continents constantly colliding, producing changes in ocean circulation. Those will obviously affect water temperature and the availability of food. And so when we, in some areas we'll see, sometimes the environment will be quite good, then something will happen and that will cause the environment to become harsher. And so we obviously changes in response to those changes in the environment as well. They're reflected in the fossil record. And so we need to take that into account as well. All right, so what's going on in the Cambrian? Well, the Cambrian was the period during which many new body plans formed. Essentially, these are plans that affect us today, even humans. And we begin to see animals moving into environmental niches. So we don't have we have some animals that obviously are quite broad. They can cover a range of different environments, and then we also begin to see the appearance of some animals that can live in only very, very restricted environments. So it's unsurprising that the Cambrian period experienced the highest percentage of evolutionary experiments, so new species, compared to any of the other, any other time in geologic history. Essentially, this is the moment where life goes, right, I'm going to see what I can do. And and evolution begins to produce a whole range of different organisms. Some of them are successful, the vast majority are failures. So although all of the invertebrate groups were present in the Cambrian, many were only represented by a few species. So there wasn't a huge diversity of organisms in each of the groups. Okay, that's purely because life hadn't had time to develop a broad range of organisms in each group. So each group only consisted of a few distinct organisms. So in the Cambrian, we have the echinoderms, so the starfish and sea urchins, we know they're about. We have trilobites, we have inarticulate brachiopods, so that's due to the hinge and tooth and socket arrangement, so we'll cover that in just a few slides. Excuse me. And we also have the archaeophytes. So the archaeophytes have already come up. We actually touched on them when we were discussing the Paleozoic Earth history, and we were discussing the formation of reefs during the Cambrian. So the archaeophytes were essentially the organisms, these cone-shaped organisms, that form the majority of reefs during the Cambrian. And these organisms actually formed the vast majority of life in the Cambrian. And you can see, there was a very broad range of organisms during the Cambrian. Some of them were relatively common. So, we know organisms like the trilobites, and then, there's going to be organisms like hallucigena down here, which we'll cover in a second, which were, very interesting in their evolutionary development, and they were, pretty much, short-lived experiments. So when it comes to life in the Cambrian, trilobites are by far the most prominent member of the Cambrian marine community, and they comprise approximately 50% of the total fauna, so the total fossils that we have. Now, most trilobites were mobile and they were benthotic, so they would have lived on the seafloor. They might have also, in some cases, dug down into the sediment, so they could have been infaunal. And essentially they will have, the vast majority of them, they will have crawled on the seafloor, or they might have been able to swim just above the seafloor. Now, you might notice there's an asterisk here. So why is there an asterisk there? Well, yes, trilobites do make up about 50% of all the fossils we have from the Cambrian. However, the reason for that is trilobites fossilize very, very well. So they were very common. They had hard shells which fossilize well, and they lived in environments where they also tended to fossilize. So this means that, yes, they probably made up a significant portion of the Cambrian marine community. However, they were probably nowhere near as high as 50% of the community. So that 50% value that you're seeing there is essentially biased by the fact that they fossilize so well. In terms of the archaeophytes, these organisms that essentially are forming the majority of the Cambrian reef structures during the Cambrian, well, they're sessile, so they're not moving. They're benthotic, so they're attached to the seafloor, and they are probably suspension feeders, just filtering out material from the water column. And they formed reef-like structures in the early and middle Cambrian, but by the late Cambrian, they had gone. So what we can see if we look at this table at the top here is that the majority of invertebrate groups and their stratigraphic ranges. So this table essentially is showing, right, where do the main groups make their appearance. So when we look at groups like the Protozoa, essentially they make their appearance in the Cambrian. Okay, the Nadaria, they make their appearance in, the vast majority of them anyway, the first group of them make their appearance in the Cambrian. However, most of them make their appearance in the Ordovician, so they're a bit late to the party. The Bryozoans, well, they make their appearance in the Ordovician. Brachiopods, Cambrian. Mollusks, Cambrian. Arthropods, Cambrian. Echinodermata, most of them Cambrian or Ordovician. So what you can see is a significant number of these groups have their first members appearing in the Cambrian. So one of the most important groups is the Brachiopods. So what's a brachiopod? So a brachiopod is an organism that consists of a shell that's made of two valves, and the valve essentially is the half of the shell. So if you look at this picture down here, this is a standard kind of organism you might see at the seashore. You might see one of these clam-like organisms. Each half of the shell is referred to as a valve. Okay, one valve, the bottom valve here, the top valve here. Now, the brachiopods that formed have this design. So they have two valves, top and bottom valve, and they come as the organisms, the brachiopods that formed during the Cambrian were referred to as the inarticulate brachiopod. So what does that mean? Well, when we're discussing brachiopods and bivalves for that matter, we have to think about the hinge region. So this region right here where the two valves meet. So these early brachiopods, these inarticulate brachiopods, did not have what we refer to as a tooth and socket arrangement. So think of your hip. Okay, so your hip bone, your femur, has a ball at the end that locks into essentially a depression in your pelvis. Okay, so that's essentially a tooth and socket arrangement. Both bones lock into each other. Now, in the case of this bivalve here, each halves of the shell are going to lock into each other because one half has a tooth, essentially a bit that sticks out, and the other valve will have a depression in which the tooth will sit. And that's very, very helpful because that helps to essentially make the hinge region stronger. So it makes it more difficult for organisms, for animals, for predators more accurately, to pull the two valves apart to get to the animal inside the shell. So the inarticulate brachiopods were a group of very simple brachiopods that did not have this tooth and socket arrangement. So essentially their hinge regions were just smooth, and the two valves were held together purely by the muscles of the organism. So essentially the only thing that was stopping the two valves being pulled apart was simply how big the muscles were that the organism had holding the two valves together. So another group of brachiopods were around. So these are the articulate brachiopods, and these are the organisms that did have this tooth and socket arrangement. And they, however, don't become particularly common until the Ordovician. So in the Cambrian we have lots of these brachiopods around, and most of them are the articulate brachiopods. They're the simplest form of brachiopods. So where do brachiopods live? Well, brachiopods can live in a couple of environments. They can live either on the seafloor or in the sediment. So some brachiopods have the capacity to bury themselves in the sediment for protection, and then they'll just try and grab food that's on the seafloor. With a proboscis, typically they'll stick out of a burrow. Or some bivalves will actually anchor themselves to the seafloor using a stem, and they will just open the shell during when they're in water, and they will then simply filter out particles that they grab that pass into or near the shell. So the inarticulate shells were composed of chitin, so that's an organic compound used to make insect shells, and calcium phosphate. So it might not sound that tough, but this combination of chitin and calcium phosphate actually produces very, very robust, strong shells. So one of the things is, how can we tell the difference between a bivalve, which is one type of clam over here, and brachiopods? Well, it's all to do with whether the valves are mirror images of one another. So let's have a look at this bivalve here. So look at these two valves. You'll notice they are pretty much mirror images of each other. So valve 1 and valve 2 are more or less carbon copies of one another. Now, the fact that the valves are mirror images, copies of one another, tells us that this organism has to be a bivalve. And now this works in about 90% of cases. Now let's come over here and have a look at the brachiopod. What you'll notice is when you look at the brachiopod, you will see that the two valves are distinctly different. One valve is larger than the other, and so that's what tells us we're looking at a brachiopod here. Once again, 90% of cases, if the valves are different sizes, then you're looking at a brachiopod. So let's look at an actual example here. You can quite clearly see that when we look at this organism, well, both of the valves are going to have this kind of semicircular shape to them. However, this top valve is going to have this concave shape here, this concave ridge running down the middle of it. And obviously this lower valve has to also have a concave shape to kind of latch into it. But it's not going to be the same shape. Okay, so each of the valves for this organism are going to be different shapes, therefore we're going to classify it as a brachiopod. So these brachiopods are very, very important during the Cambrian, and they're going to make up a very significant portion of the Shelly marine fauna around at the time. So what about soft-bodied organisms during the Cambrian? Well, as we've already touched on, when it comes to fossilization, fossilizing stuff that has a soft body, so no skeleton, is very, very difficult. And so this means that the fossil record that we have is obviously dominated by organisms that have exoskeletons because they fossilize so well. So we need to remember that the Cambrian was a period of evolutionary experimentation. However, most of the roads were dead ends, so most of the experimentation that took place didn't really go anywhere. So, as the Sauk Sea transgressed over Laurentia, a portion of the Cordilleran Shelf, so essentially what is now the modern-day west coast of North America, specifically an area of what is now British Columbia in Canada, went from an early Cambrian sandstone, so coastal deposit to a middle Cambrian shale. So it was essentially, the water depth went up, so sea levels were rising. Now, it just so happens this area of British Columbia, the muddy area, the muddy facies that it was in, became anoxic. So it became oxygen poor. So that means that any organic material that fell into it could not decompose, because decomposers require oxygen. And so this means that soft-bodied organisms that fell into that environment would fall onto the seafloor, and then their carcasses would be covered over by sediment, and essentially their carcasses could be, essentially the impressions of their bodies at least, could be retained within the rocks. So this is very, very important. So this means that later on, when geologists come along, we can crack open the rocks at the Burgess Shale, and we will see the impressions of these organisms. Now, this is a very rare situation. This isn't common at all. This is a very lucky situation that we're in. Okay, and we are also extra lucky that this particular environment existed in the Cambrian, because it allows us to have a look at these organisms from the very, very early Cambrian that we otherwise would not be able to look at because they are soft-bodied. So here's a few examples. So one of the first examples is Hallucigena. And Hallucigena is an extremely interesting organism. So you can see, we have these spikes. Well, to begin with, initially we didn't know whether it walked on the spikes or whether the spikes were on its back. And then up until recently, we weren't actually sure which end was the head and which end was the rear. So, science in action took us a while to work that one out. And we now know that it had legs on the bottom, which it used to move around, spines on the back for protection, and now we're pretty certain which end was the head and which end was the rear. So then we also have other organisms like Canada spinosa, and as you can quite clearly see, it seems to be some kind of arthropod, and you can see the shell itself has this very feathery appearance to it. You can see that reflected in the fossil itself. So essentially, this is the head region here. You can just about make out an antennae here, an antennae here. You can see this is the line of the body here, and these essentially are the portions, the segments of the exoskeleton either side, covered in these feathery appendages. So then we have an organism like Morelia splendens, and in this case you can see this one is relatively easy to identify. It looks very much like a member of the arthropod family. It looks a lot like an insect, doesn't it? So this one is more easy to place within a group of organisms. So once again, though, in terms of the material it's made from, this is not a hard shell. So it's not a calcium carbonate shell. This is an organic shell that otherwise would not have fossilized. So this is an organism that we otherwise wouldn't know about if we hadn't gotten lucky and it hadn't fallen into this this Burgess Shale region. So then we have worms fossilized. Once again, worms, we might have found the fossilized burrows, but we wouldn't have found the fossilized worm because it's a soft-bodied organism. And so this particular worm is, now I'm absolutely going to murder this particular pronunciation, Otia prolifica. And you can see it has this long segmented body, and you can see at the top here it has this proboscis, and so it would have had this, we believe it would have had this U-shaped burrow with the head at one end, and the head would have sporadically popped out, and it would have tried to grab food from the sea floor. We don't think it was a filter feeder. We think it would have actively tried to grab organisms as they scuttled across the seafloor. So then we have an organism here, Wapita philensis, and you can quite clearly see now this one is very clear in its appearance. You probably saw this and you thought, right, it's a shrimp. And yeah, chances are it is probably related to modern day shrimps. But once again, the shell of the organism itself, just like a shrimp now, the shell is made of organic material. That would not have fossilized otherwise, so we wouldn't have known this organism existed. So these are precursor organisms to modern day shrimp by the looks of them. And then we have other organisms like Micrometra bagenesis. Now this one is a brachiopod. Okay, we can see it had a stalk that anchored it to the seafloor. You can just about make it out in this diagram here. Now, this one isn't particularly interesting in terms of what it is. It's a brachiopod, it's relatively common, relatively unexciting. What is interesting, though, is the fact that we managed to preserve the little appendages that were coming out, that were grabbing small particles of food in the seawater and then pulling them back into the shell and dropping them essentially into the animal's mouth/gut, where they could be digested. So in this case, it's not really what the animal is that's interesting, it's what it shows us about how it actually caught its food that's interesting. So it was showing us that, brachiopods and bivalves in the Cambrian were essentially, getting their food in the same way as modern brachiopods and bivalves do now. So the Burgess shell is not only important due to the fossils, but it also gives us information about the phylums that were present. So the basic model for the Cambrian explosion is that the modern phyla were established in the Cambrian, be it with fewer organisms in each phylum. So each of the groups, the main groups of animals that we have in the modern environment, they made their first appearance in the Cambrian, although as we've already discussed, there were few organisms in each group. And the Burgess shell is helpful because it allows us to add more organisms to each of these groups. So we not only have the organisms in each group that had the exoskeletons, we also have the organisms in each group, well, we also have some of the organisms for each group that were soft-bodied as well. So following this argument, the number of organisms in each phyla has been increasing through time, but the number of basic body plans has remained constant. So essentially what we're seeing here, especially if we go back to this organism here, you can see the basic body plan here is remarkably similar to a modern day shrimp. So what it's showing you is, most of the basic body plans were established in the Cambrian and they've been carried on through to the present day. So, however, using early Cambrian fossils as evidence have suggested the explosion marked a period of experimentation and extinction for many phyla. So yes, the early Cambrian, the Cambrian explosion, was a time where we had lots and lots of organisms forming. However, the vast majority of these organisms never went anywhere and they became extinct. So let's look at a couple of organisms that are actually quite difficult to place within the modern phyla. So right, I'm really going to murder this name, for which I do apologise. Vetriculata? Nope, let's try that one again. Okay, here we go. Vetriculata. And you can see this organism appears to have been a segmented, almost worm-like organism of some kind. It appears to have had a fin-like feature at the end, probably for locomotion, so it was probably actually capable of swimming through the water column. It appears to have had a mouth at one end, and apart from that, it's pretty much anybody's guess what was going on. You can actually see a fossil of it here. So this is going to be the mouth end, the front end here. This is the main body here, tapering, so essentially it's tapering to this mid-region point here, where it balloons out, and we have part of the tail structure here. The bottom of the tail structure is missing. And at the bottom here we have a fossil of an organism called Wuaxia. And you can see once again, so you can see it had several large spines coming off it, and it had an exoskeleton consisting of numerous plates. Now, this organism on the whole has been incorporated into the group Echinodermata. That's the group that includes sea urchins, and that's where it sits now, but it doesn't fit comfortably into that group. So we have to remember that the vast majority of organisms that we see in the Cambrian, and especially in the Burgess shale, can be put into modern groups. However, some of them cannot. So the model proposes that the basic body plans that the phyla have today are the result of multiple variations, with the survivors making up the modern phyla. So these body plans appear in the Cambrian, and evolution essentially has carried them forward through each generation, through each new essentially evolutionary step, all the way through till the modern day. So this means obviously there could have been phyla during the Cambrian which did not make it to the present day, and that would explain, certain slightly interesting organisms like this one here that don't fit comfortably into any of the modern phyla, and obviously that would suggest that that particular phyla died out. So as the number of early Cambrian fossil sites increases, many of these strange organisms have been successfully assigned to modern phyla. However, this argument is not yet resolved. So there are some geologists that say, well, no, clearly all the modern phyla were established in the Cambrian, and that's it. There were no, there were no extra phyla present. And there are some geologists that say, yes, the modern phyla were established in the Cambrian, but there were also some other phyla that didn't make it through to the present day. Okay, so we've been going for an hour. This is a good place to pause the presentation, get up, have a walk around, go in a glass of water, and please come back in a few minutes. Okay, so we made our way through the Cambrian. We had all this experimentation going on. We had, not only the hard-bodied organisms, but courtesy of the Burgess shell, we also know we had all these soft-bodied organisms. And so, as we move from the Cambrian into the Ordovician, we begin to see some essentially increase in the diversity of the majority of the phyla that make it through the Cambrian. So, if these phyla, these groups of organisms, make it through the Cambrian, essentially we begin to see an increase in the number of organisms in each of the phyla that make it. So the Tippecanoe transgression created a large warm sea during the Middle Ordovician, and that opened up new marine habitats. This is in reference to Laurentia, but also other parts of the world. Essentially, the Tippecanoe transgression was caused by rising sea levels, and so it not only occurred, Tippecanoe age transgressions not only occurred on Laurentia, but also over other continents. So there was an opening up of these warm, shallow seas during the Ordovician. So the Ordovician community displays increases in the diversity of the brachiopods. We see increased radiation of many invertebrate phyla. So we see the number of distinct organisms increased. We see corals or bryozoans, pictured behind. We see increases in abundance and diversity of the cripes, another word that I'm about to muck up here. Erectarchs, a type of phytoplankton, and we see archaeophytes are out and reefs are beginning to be made from stromatophoids, bryozoans, and tabulate and rugose corals. And now these aren't long reefs like the kind of reefs you think of now. So the archaeophytes form these long linear reefs. The type of reefs we get in the Ordovician are what we refer to as patch reefs, the little kind of mounds essentially. So we see a massive increase in diversity of all Shelly fauna, and that's referred to as the Great Ordovician Biodiversification Event. Essentially, that event is an increase in the number of organisms in the phyla. So as we make it into the Ordovician, what do we see? Well, we see, number one, the trilobites have made it through the Cambrian. We have the echinoderms are still around. We have the brachiopods still around. We have the cephalopods well established. We have the first fish making their appearance. Now, those will obviously be discussed more in the vertebrate portion of this presentation. We have the crinoids, these organisms, excuse me, making their appearance in the Ordovician and becoming very well established. And then we have some more interesting species like the Eurypterids. And do you know what? I'm honestly not sure what that one is there, making their appearance as well. So the Ordovician is essentially interesting in that, well, it's limited in terms of its interestingness by the fact that what we see is just a continuation of the species that move from the phyla that move from the Cambrian into the Ordovician. So there's no significant change. All we see is just an increase in the number of organisms in each phyla. Now, the Ordovician is interesting, though, because there are several fossil groups that provide extremely useful biostratigraphic correlations make their appearance in the Ordovician. So the first group is a group we've already discussed. It's the articulate brachiopods. So these, remember, the brachiopods are the shelly organisms that have two different sized valves, and they have the teeth and socket hinge region. So that makes the shell more difficult to pull apart. Now, they become dominant in the Ordovician, and the inarticulate brachiopods essentially become the minority of the brachiopod species around. And of course that makes sense. The articulate brachiopods have a better shell design, and so of course they're going to have an evolutionary advantage. So that's not really a huge shock. The one thing the articulate brachiopods do, though, is they absolutely explode. They become, inarticulate brachiopods were common in the Cambrian, but by the time we make it into the Ordovician, the articulate brachiopods are just everywhere. And so that makes them really great fossils for doing biostratigraphy, because they're very geographically widespread, and there's lots of species that are relatively short-lived. In the Ordovician, we also see a group of species appearing which are called the graptolites. So they're planktonic organisms, so they floated in the water, and they are millimeter to centimeter sized colonial filter-feeding organisms. And most of the species in the graptolite group lasted less than 1 million years. And so they had a very, very short geologic range, which is great for doing stratigraphic correlations. And they were most common during the Ordovician and the Silurian. They're not around now, they went extinct eventually. The other thing is, though, is that because they lived in the water column and they were free floating, well, it meant once again they had a very, very broad geographic distribution. So they're absolutely great for doing biostratigraphy over very, very large distances. And actually, we can see you have a picture of a couple of these graptolites behind here. Okay, you can see essentially they have this tooth-like pattern, these particular species anyway. And each one of these teeth would have represented the location where a single organism would have lived, essentially, and essentially filtered food out of the water column as the larger structure floated along. The final organism that's very, very helpful for biostratigraphy from the Ordovician onwards, or at least in the Ordovician, are the group called the conodonts. And they are simple, microscopic eel-like fish. Now, their bodies do not fossilize. Okay, their bodies are made from, organic material, and the skeleton is made of cartilage, just like a modern shark. So the skeleton doesn't fossilize. Just like a modern shark, what does fossilize, though, are the teeth. So these teeth are commonly referred to as conodont fragments. And so what you can do is you can take a rock from the Ordovician, and you can crush it, you can sieve it, and you can extract the teeth from that rock, and you can identify the individual conodont species using the teeth. And once again, conodont species were very widespread, and each individual species were typically relatively short-lived, so they are absolutely excellent for stratigraphic correlations. So the end of the Ordovician was marked by a mass extinction, possibly due to extensive glaciation on Gondwana, decreasing sea levels and reducing surface water temperatures. So, we have all these beautiful kind of, Ordovician aged shallow marine environments, and then all of a sudden, as you go from the Ordovician into the Silurian, you get massive glaciers forming on Gondwana. Remember, Gondwana sat over the South Pole at this point. And all of a sudden, global sea levels drop, so all those nice shallow water marine environments, they suddenly disappear. And at the same time, because you have all this global glaciation, that's going to reduce global temperatures. So ocean surface temperatures are going to decrease. That's going to cause a decrease in the amount of available food, because the numbers of photosynthesizing organisms is going to decrease as the surface water temperature drops. And so essentially, this is going to lead to a collapse of the marine ecosystem, and we see that as a mass extinction. So we see lots of organisms dying out at the same time. And to give you some kind of idea, approximately 50% of brachiopods and bryozoans died out in North America alone during this period. So as the Tippecanoe Empiric Sea retreats, we see about 50% of brachiopods and bryozoans dying out. So it was a pretty big extinction event. So we make it through the Ordovician, and we're now into the Silurian and the Devonian. So what's going on now? So after this mass extinction that marks the Ordovician-Silurian boundary, we see the brachiopods, the bryozoans, the gastropods, the bivalves, the corals, the crinoids, and the graptolites all rediversify. So they all take a bit of a beating. They all lose members of their phyla as you transition from the Ordovician into the Silurian, but they make a comeback. And they rediversify, and they take over their environmental niches that they had before, and in some cases they even take over new ones. So we see marine transgressions leading to the formation of shallow seas once again. So now we're into the Silurian heading to Devonian. We're kind of on the verge of the Kaskaskia. So we're going to see a steady increase in sea levels, which is going to lead to the Kaskaskia marine transgression over Laurentia. Now that's mostly focused into the Devonian. So the marine transgression led to the formation of shallow seas. Once again, shallow seas great for reefs. And so what we have now is we have huge areas of passive continental margin, shallow, warm seas, very, very productive, and so it's going to be great for life during this period. So what we noticed during this period is the patch reefs of the Cambrian Ordovician were replaced with large reef structures. So once again, we're back to these very long linear reefs. We see a diversification of reef-building fauna, and we see especially the appearance of the rugose corals. And the rugose corals are also referred to as the horn corals because they have this very, very distinct horn-like shape. Okay, and they can exist individually or they can exist as colonies. So the reef developed a modern morphology. They were constructed of stratopoids, which made their appearance in the Ordovician. Tabulate, you can see this is a tabulate coral here, and rugose corals, these horn corals down here. And the rugose corals especially become very, very dominant features of the Silurian and Devonian reef communities. Just like the Archaeopsyphids became the dominant species of forming Cambrian reefs, as we move into the Silurian and Devonian, the rugose corals become very, very dominant in terms of the reef communities during that time period. And so as we move into the Silurian Devonian, what do we see? Well, we see that the fish become more better adapted. They become more advanced. We see the Eurypterids are still hanging around. We see the crinoids are still here. We see the trilobites are still around, but their numbers have significantly decreased. The trilobites are steadily on a downward trajectory from about this point on, unfortunately. And on the whole, everything is pretty much as you would expect. And we can also, at this point, you can see from this picture, we also see that life has essentially made it onto the land by the Silurian. So one of the more interesting organisms that really establishes itself in the Silurian into the Devonian is the Eurypterids, and they're an arthropod. And they made their first appearance during this window, and they have this very distinct scorpion-like body. You can see, so their body essentially is very broad, it's segmented, and it tapers off into essentially a tail that ends in a pointy structure. Okay, hence the fact they're sometimes referred to as sea scorpions. Now at the front, you would often have a pair of pincers for grabbing things, grabbing food. And typically you would have a very well-developed set of limbs which would be used for propulsion, to actually push them through the water. Now, Eurypterids are very interesting in that not only are they very widespread, so they were very successful creatures, but they are also one of the first species that we know actually managed to make the transition from saline water into brackish water, so that's kind of like semi-saline, and then finally into freshwater environments. They're one of the first species that actually makes it from the oceans, and we know they made one of the first species that we know made it from the oceans into freshwater environments. Now that's important because when we think of how life made it out of the oceans onto the land, the general rule of thumb is that life has to go from the oceans into essentially the freshwater environment and then onto land. It would be very difficult for life to go straight from a salty marine environment straight onto the freshwater environment that you have on land. Life would have been, that would have been extremely challenging. Whereas transitioning from marine to environments like rivers, and then from rivers onto land would have made a lot more sense. So Eurypterids are extremely successful and they're very, very widespread. The Silurian Devonian period is also where we see the appearance of the ammonites, which is a group that I'm sure most people are familiar with. So they're a group of cephalopods, and they have this very, very distinct spiral morphology. Okay, now they developed from the nautiloids. The nautiloids are actually a group of organisms that are still around today, and they appear during the early Devonian, and they very, very rapidly diversify. They become very common. So they also become very successful. They become arguably the dominant organism in the water column. And we see they are excellent guide fossils between the Devonian and the Cretaceous, where they are, as I said, extremely widespread, extremely common, lots of different species, most of them are relatively short-lived, and so that means they're absolutely great for doing biostratigraphy. And we also see that the patterns on the shells, the suture marks on the shells, change over time, and so that becomes very helpful for dating purposes as well. And so they become a very, very common predator in the water column throughout the late Paleozoic and into the Mesozoic. And of course, they're eventually killed off by the Katy impact that also killed off the dinosaurs. So the end of the Devonian was marked by another mass extinction. And this extinction did substantial damage to reef communities. However, land plants appear to have been relatively unaffected. So whatever happened was focused mostly in the oceans. So furthermore, marine communities in tropical regions were more affected than those in more temperate climates. And so this leads to a relatively straightforward conclusion about what was causing this mass extinction. So this suggests that decreasing ocean temperatures were to blame, as this would primarily affect tropical reef communities. So obviously what happens is, as the poles get colder, well, that means that, that obviously the water towards the poles will decrease in temperature. Now this means that the temperate regions will shift to the south, they'll move towards the equator. So this means that any organism that lived in a temperate region can just follow that temperate region as it moves south. So organisms from these temperate regions aren't really that affected by this mass extinction. Problem is, though, is as the temperate regions move south from both the North Pole and the South Pole, so as they move towards the equator, should I say, from both the North Pole and the South Pole, well, that means the tropical region, the equatorial regions, suddenly finds itself being squished. So the equatorial regions, the equator, the equatorial warm water regions suddenly find themselves getting compressed from both the north and the south. This equatorial band suddenly gets squished, and it's no longer as big. And so this obviously means that all of a sudden you have a lot of organisms that are trying to squeeze themselves into a much smaller area of the world's oceans. That's going to lead to, greater competition. It's also going to lead to stressing of the food chain. And so obviously that means that it's far more likely you are going to get collapses of the food chain. And so that's obviously going to cause mass extinctions as the food chain starts falling apart. So communities were also affected by the closure of the Iapetus Ocean and the orogenic vents that are associated with it. So as Baltica is heading towards Laurentia, that means you have nice passive marine, that means you have nice boring passive marine margins are about to be destroyed, and they're replaced by active margins, which are nowhere near as nice for life to live in. So as we move into the Carboniferous and the Permian, what's going on? So, as with previous mass extinctions, marine life rapidly recovered in the early Carboniferous, and we have renewed adaptations and rediversification. So, once again, all the phyla, the modern phyla, are present at this point. They've been increasing in diversity throughout the Silurian and the Devonian. You have this mass extinction at the end of the Devonian caused by changes in sea temperature, and then all the phyla make it through. And then once we're into the Carboniferous and Permian, all those phyla then rediversify again. So we see an increase in the number of species in each phyla as they exploit new environments. Oh, by the way, while I remember, today's code word is coffee. I repeat, it is coffee, as in the drink you have in the morning. So ammonites and brachiopods assumed important ecological roles during this period. Okay, so we see the ammonites are, arguably the dominant organism in the ocean column, in the seawater itself, and we see the brachiopods become extremely important on the seafloor. Other groups such as crinoids and bryozoans steadily increase in diversity, reaching their peak in the Permian. So the crinoids, essentially this organism we see behind, we've seen them in previous pictures, they, like most organisms, they take a bit of a beating at the end of the Devonian, but as we head into the Carboniferous and the Permian, their numbers recover and they rediversify. The same goes for the bryozoans. We see stromatophoids, tabulate corals and rugose corals never recover from the mass extinction at the end of the Devonian. And so at this point, we move back to a patch reef morphology. So we lose these long linear reefs, and we're back to a morphology where reefs occur as these little isolated mounds, essentially. And the reefs that actually formed during this period are mostly made up of crinoids, a group of organisms called blastoids, bryozoans, brachiopods, and calcareous algae. So you're not really seeing reefs that are being made up by, coral-like organisms. So the reefs that are forming at this point are essentially just piles of dead crinoids. So crinoids begin to occur in very, very large numbers, and obviously as they die, they they deposit lots and lots of material onto the seafloor, and over time this material piles up and up and up, forming these reef mounds. So the carbonate remains from these bryozoans and these crinoids produced massive amounts of skeletal remains, and so all of these skeletal remains end up producing massive bedded limestones during the early Carboniferous, and they really are absolutely stuffed with these crinoid fossils. It's very, very easy to identify a Carboniferous limestone because it is absolutely rammed with bits of crinoid. So Permian communities were similar to Carboniferous communities, but on the whole they were smaller. Why were they smaller? Well, as we move from the Carboniferous into the Permian, we begin to see a reduction in the amount of continental shelf space available. So during this period, what's going on? Well, we're seeing Laurasia is moving towards Gondwana, and there is, the collision has begun. And so this means that all those nice passive continental margins between Laurentia and Gondwana are being destroyed. So there's a decrease in nice passive margins. We also see at this point that Siberia is moving towards Baltica. Eventually that's going to form the Ural Mountains, and once again that's reducing the amount of passive continental margins around, so there's less space for organisms to live in. And so this means that organisms do a couple of things. Number one, the communities tend to lose organisms. There tends to be a decrease in the diversity. And organisms also tend to become smaller because there's increased competition for food. So during the Permian, we have a group called the Productids. They're a group of spiny brachiopods, became the dominant brachiopod species. We also see during the Permian the appearance of the fuselids, and they're a type of spindle-shaped foraminifera, and they become very common. They diversify from the late Carboniferous into the Permian, and they are very, very widely distributed, and so they're a very, very important guide fossil. So, when you try and do biostratigraphy, especially of the Carboniferous and the Permian, these fuselids, which you can see a picture of some of them here, are very, very important in terms of, trying to link up bodies of rock over large distances. So when we look at mass extinctions, we notice the following. So, we've already been through a couple of mass extinctions. So we've had the Ordovician Silurian extinction, and we've had the extinction between the Devonian and the Carboniferous. So when we look at these mass extinctions, we notice a few things. We typically notice that they affect both land and marine life. Normally, tropical organisms are more affected than organisms from higher, more temperate latitudes. And although some groups within a phyla become extinct, the phyla itself will persist. So when we look at the fossil record, we see that the Cambrian had several extinction events which dominantly affected marine invertebrates, particularly trilobites. Now that's not a huge shock, because you have to bear in mind that during the Cambrian there is no life on the continents. So obviously, when it comes to affecting life on land, well, there's nothing there to affect. So in terms of extinctions throughout the rest of the Paleozoic, we see there's an extinction between the Ordovician and the Silurian. Now that affects all invertebrates in the marine community. There's an extinction between the Devonian and the Carboniferous. That very heavily affects reef-building organisms and armoured fish. So the reef-building organisms are, of course, the stromatophoids, the tabulate corals, and the rugose corals. They don't do very well. And right at the end of the Permian, we have the Permian Triassic mass extinction, also called the Permo-Trias mass extinction. And this one is the big one. It's the biggest mass extinction. It's the closest we've ever come to just completely resetting all life on Earth. And the Permo-Trias mass extinction affected up to 96% of all marine invertebrates went extinct, and in excess of 65% of all land animals became extinct. So it was an absolutely massive mass extinction. So the Paleozoic and the Mesozoic have several mass extinctions each. And of course, the best known is the Cretaceous Tertiary mass extinction. And of course, that's the one that killed off the dinosaurs, the flying reptiles, and the marine reptiles, not to mention the ammonites as well. Although some extinctions like the KT event were relatively rapid, the vast majority of extinctions we see in the geologic record are relatively gradual. So typically what happens in the geologic record is we see the extinctions are gradual. Whatever the problem is builds up over time until we reach a tipping point, at which point things can't be recovered from. Okay, and then we get the main portion of the mass extinction occurring. It's just like a roller coaster. You go up, 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 up until you reach the point of no return, and then you go plunging down the other side. That's pretty much the basic model. So the vast majority of mass extinctions do not have a single cause. So the Cretaceous Tertiary mass extinction, the one that kills off the dinosaurs, is an anomaly. That's caused by a meteorite that's hurtling onto the surface of the Earth. That's by far and away the main reason why this happened. Nearly every other mass extinction, on the other hand, has a range of environmental factors which all contribute together to lead to the mass extinction. And so, that means they are very, very difficult. It's very, very difficult to work out exactly what the cause was for a lot of these mass extinctions, because there's so many competing causes. So this brings us on to the Permian-Triassic mass extinction. So as I said, the numbers of organisms lost are pretty huge. So 96% of all marine invertebrate species became extinct. 50% of all marine invertebrate families became extinct. 70% of terrestrial vertebrates became extinct. Greater than 65% of all land animals became extinct. And 33% of insects became extinct. So that's pretty serious. So rugose corals, tabulate corals, fuselids, and several bryozoans and brachiopod families, they're gone at this point. They don't recover. So the cause and duration of the extinction remain uncertain. So geologists fall into two camps when we discuss the Permo-Trias mass extinction. There's the catastrophic group, and they say whatever caused the extinction occurred rapidly. So think a meteorite impact. And then there's the gradual group. Their argument is that whatever caused the mass extinction built up steadily over time until we reach this tipping point, this point of no return, at which point the main part of the extinction occurs, and the extinction itself is also quite a gradual event. So the catastrophic group say, note, the extinction event happened very, very quickly, whereas the gradual group says, no, the extinction event had a gradual build-up, and the extinction event itself was also quite gradual. So geologists have to explain why the extinction event, this Permo-Trias mass extinction, had such a long recovery time. So typically when there's a mass extinction, we see life recovers pretty quickly. Okay, so life is relatively hardy, and when there are all these environmental niches that open up because of an extinction, we see life will often very rapidly diversify and fill the niches that are left. However, with the Permo-Trias mass extinction, that's rather different. We see that it takes about 4 to 6 million years before diversity actually begins to increase again. So that's a pretty serious lag time. So something's going on that's inhibiting the capacity of life to essentially take back environments from which it's disappeared. And we also see that it took up to 30 million years for terrestrial vertebrates to fully recover. So it took about 30 million years for the life on the continents, so the vertebrate life on the continents, to actually recover from this mass extinction. Once again, this is a very long lag time between the extinction event and the recovery. So something is going on that's clearly causing a very, very slow recovery. Another thing we need to explain is why the mass extinction seems to have predominantly affected organisms that have a calcium carbonate shell. So, as we discussed, shells can be made from calcium phosphate, from silica. Those organisms suffered, but they didn't suffer as badly as organisms that had a calcium carbonate shell. So there's got to be some reason for that as well. So we have to try and explain all of these things. So what else do we know about the Permo-Trias mass extinction? Well, we know that it's marked by a decrease in the carbon-13, carbon-12 ratio. So life typically concentrates carbon-12. So that means when there's a significant decrease in the amount of life on Earth, that means there's lots of carbon-12 being released back into the environment because there are lots of animals that are dying and their bodies are decomposing. It also means because it's an extinction, there are fewer animals to actually reuse that carbon-12. So it's essentially going back into the environment and it's not being taken back out. So we see the amount of carbon-12 go up, and so that affects the carbon-13, carbon-12 ratio. What else do we see? Well, we see there's probably an increase in global temperature of around 8%. 8 Celsius. We also see an increase in global carbon dioxide. So essentially we see an increase of about 2,000 parts per million. So to give you some idea, pre-industrial revolution concentrations of calcium carbonate in the atmosphere were about 200 parts per million. So, that's a pretty significant increase. That's going to cause some very serious global warming. So the amount of carbon dioxide increasing ties back into this increase in global warming that we see. We also see indications that there's an increase in the amount of ultraviolet radiation making it to the surface of the Earth. This suggests that something is going on with the ozone layer. We see there's a spike in marine and terrestrial fungi, and that one's relatively straightforward. That one's due to the large amount of dead plants and animals, and obviously fungi they're decomposers, they break down these animals. So the fact that we suddenly see a large increase in the amount of fungi tells us we've got lots of dead animals around and lots of dead plants. And we see the continents becoming more arid. So we have to be able to explain all of these things that we see. So this is our challenge. So are there any causes that we can reject when it comes to the Permo-Trias mass extinction? Well, to begin with, we'll need to take a quick break now because we've been going for over 20 minutes. So let's pause the presentation here, get up, have a walk around, and please come back in a few minutes. Okay, so are there any causes that we can reject for the Permo-Trias mass extinction? Well, we can reject a reduction in the size of the continental shelf environment. So as I was saying, as we were going through the Carboniferous and the Permian, we've been steadily seeing a reduction in the amount of nice passive continental shelf environments as the continents are colliding into each other. Now this would have put a stress on marine life. However, the majority of those collisions were complete by the late Carboniferous. So that means any adjustment would have occurred before the Permo-Trias boundary. So that means that it's unlikely that the loss of these passive continental margins were the cause. Now the boundary does mark a period of low sea levels, but whether that's important is uncertain. We can also reject glaciation as the cause of the mass extinction. Why is that? Well, glaciations had been occurring all the way through the Carboniferous, and they had actually been waning as we headed into the Permian. So that means that glacial events were actually becoming less frequent and less severe. So that would mean that on the whole, the chances of glaciers being the cause is significantly reduced. So what's left? Well, the obvious one that we're going to go to straight away is a meteorite impact. So it would help to explain the rather rapid extinction, but as it happened 250 million years ago, the evidence for any meteorite impact would be very difficult to find, especially if it happened in the ocean. So we need to remember that oceanic crust is typically recycled on a 200 million year timescale. So that means there's a very good chance that any impact crater caused by the Permo-Trias, the impact crater produced by an impact at that time, has been subducted and lost. So that means it's going to be very difficult to find conclusive evidence. It also means that if the impact crater happened on land, well, obviously that means the crater is going to be filled in by later sediment, and so it may be very difficult to spot. So although there's no impact crater of sufficient size and age that has been found so far, there is a possible candidate that does exist. It's an impact crater in Argentina. I'm not even going to try and pronounce it. Now, it's too small to have actually caused a global extinction on its own. However, it just so happens that it's situated in an area where the rocks are very oil rich. So what that could mean is if an impact did occur, it could have caused substantial quantities of oil and gas to essentially burn. This could have led to the release of large quantities of carbon dioxide, or in the case of gas, it could have led to the release of large quantities of methane. And so this could have put significant amount of carbon dioxide and methane into the atmosphere, possibly leading to global warming. Now, there has actually been another study in 2017, and that's actually found a spherical feature on the seafloor around the Falkland Islands. So that's right at the southern tip of Argentina. And this crater-like feature is about 200 to 250 kilometers in diameter, and that would make it larger than the Chicxulub impact crater. That's the one that essentially killed off the dinosaurs. So now it's very, very sketchy whether this is the right age. So the seafloor in that area is probably less than 250 million years old. So it's unlikely that if it is an impact crater, it is responsible for the Permo-Trias extinction, but that's also something that's being monitored. So we need to keep an eye out for that one as well. But for the time being, an impact, well, unfortunately, chances of us finding the evidence of it relatively low, so we're going to have to go with uncertain / probably not. Okay, so option number 2, stratified global ocean. Okay, so what's a stratified ocean? Well, a stratified ocean is simply when the ocean splits up into distinct layers. Now, these layers that form in the ocean can, on some occasion, actually have difficulty communicating with one another, so they can't swap water between the layers easily. So this means that the layer at the top, which is the one in contact with the atmosphere, can be very, very oxygen rich, but obviously if it's not swapping oxygen rich water with the layer below, well, that means any layer below the top layer is going to be relatively oxygen poor, as the oxygen is going to get slowly used up, and eventually it's going to become anoxic. So the stratified global ocean model says that the oxygen-rich surface waters stopped mixing with deeper waters in the oceans, and this means that carbon dioxide concentrations in deeper water increased, leading to deep sea anoxia. So all the oxygen-using organisms that use oxygen to respire in deeper waters, well, obviously they started to use up the oxygen, but it wasn't being replaced, and so eventually you get to the point where the oxygen becomes exhausted, at which point these deep waters become anoxic. So the lack of circulation between the top waters and the lower layers would also have allowed the water to become stagnant. So, so I'm sure if you leave, a glass of water somewhere and, it's still, and it's typically if it gets heated up as well a little bit, then often the water will become a bit funky. And, essentially that's the basic principle as well. The entire ocean would have become a bit, funky. So one of the other problems is that if these oxygen-rich surface waters are not mixing with colder water from below, well, that means the layer at the top is getting warmer and warmer and warmer. That means essentially it's becoming more and more buoyant. It wants to rise. That makes it even more difficult for this top layer of water to mix with any kind of denser, colder water below. So that also helps to inhibit the possibility that this top layer will mix with the layers below it. So that's going to also increase the chance of there being some kind of deep sea anoxic event. So as you can probably guess, this heating up of this top layer of water, well, that would tie in very nicely with some kind of global warming situation. So these pair of diagrams here kind of shows you relatively nicely the kind of situation that you could get. So this diagram here shows you the ocean water temperature for a region of the Earth. I believe it was somewhere near Indonesia. And you can see during the winter months, you can see there's a relatively homogeneous profile. Okay, what you can see is as the water depth gets shallower and shallower and shallower, the temperature gets lower and lower. Well, that's understandable because there's less water. As the water gets shallower, it's going to be more and more affected by the temperature of the atmosphere. So, that's not really a huge shock. Now, notice the summer temperatures on the other hand. So what you'll notice is the top layer of water here is getting heated up by the sun, and so it's becoming very, very hot, surface temperature 22 Celsius, and so it's becoming very, very buoyant. And so what you can see is there's quite clearly a dividing line right here that separates this hot top layer from this lower cold layer. Okay, so this is the basic situation. So in this data, we have a clearly stratified situation. We have this top warm layer of water here, and this lower cold layer of water here. And if these two layers are not communicating very, very well, so if the oxygen-rich water here isn't being transported and mixed with the more oxygen-poor water down here, well, that means this lower layer of water can become steadily more and more oxygen poor, and if things went to the extreme end of the scale, it would eventually become anoxic. Okay, so if the water down here becomes anoxic, well, that leads to an oceanic anoxic event. So as we discussed, that means the oxygen-rich water up here is not mixing with the water down here, so this water down here becomes essentially oxygen poor. It all gets used up. So obviously that means animals die, and that means the organic material will just fall onto the seafloor and it won't decompose. Now, on the face of it, that's not terrible. The problem is this. If it's not decomposing, well, some organisms are going to come along and they're going to try and consume that organic material. And the organisms that are going to come along and try and do that are sulfate-reducing bacteria. So this is a group of bacteria that instead of using oxygen to respire, they use sulfur. So they can survive in oxygen-poor requirements because they don't need oxygen. And so what they're going to do is all of a sudden they're going to find an environment where there is a pile of food around and no competition. And so they're going to start decomposing all this organic material that's on the seabed. And as part of the process of using sulfur to respire, they will produce hydrogen sulfide as a byproduct. Just like we produce carbon dioxide, they produce hydrogen sulfide. And so what's this going to mean? Well, some of that hydrogen sulfide will mix with ocean water, seawater, and that's going to produce sulfuric acid. And that could cause the pH of the oceans to decrease. There's also the possibility that some of that hydrogen sulfide gas makes it through the oceans and into the atmosphere. And if that happens, that's going to combine with water in the atmosphere to once again produce sulfuric acid, and that's going to lead to acid rain. So the end result of both of those situations is it could lead to a decrease in the pH of the oceans, and it could lead to a decrease in soil pH. As that acid rain hits the Earth's surface and mixes with the soil, it can cause the soil pH to drop, and that can kill the roots of plants. And obviously, if the root of plants dies, if the roots of a plant die, so does the plant. So, as you can see, this kind of situation, if we have an oceanic anoxic event combined with all these sulfate-reducing bacteria, could lead to very serious repercussions. So, what else? Well, we've already touched on global warming. So, the formation of a stratified ocean is made easier, essentially, if we have a warm global climate, because that means this layer of water right at the top of the oceans becomes very, very hot, therefore much, much more buoyant, and therefore it's less likely to want to sink and mix with the colder water below. So, if we have global warming during this period, that could possibly explain why we have this stratified ocean happening. Another thing is that global warming could also have very serious repercussions for the movement of air in the atmosphere, and so that would have had an effect on global weather patterns. So imagine if you're an organism and you're living in an environment that's typically, relatively warm and relatively wet, and then all of a sudden, due to these changes in atmospheric circulation, your environment goes from being very warm and wet to suddenly being very hot and very dry. Well, obviously that means you're in a lot of trouble. And so, if there are changes in atmospheric circulation patterns, that could also have contributed significantly to the extinction of organisms, especially on the land. So, in terms of global warming, it could have actually occurred due to the next 3 points. So the next 3 points could possibly have led back to global warming. And global warming could then lead us on to a stratified ocean. A stratified ocean then leads us on to an oceanic anoxic event. So you're beginning to see how essentially one cause could possibly snowball and lead to other things happening that eventually leads to an absolutely catastrophic extinction event. So the next possibility is gas hydrates. So the gas hydrate model says we didn't have a stratified ocean, but it says the climate starts to warm up, and so we start forming warm water at the top of the ocean column. And in this model, it says that this warm water can circulate with the waters at the bottom. So that means warm water gets pulled down and it mixes with the water down here, and so it makes it a lot warmer. So you have very warm water coming down, producing warm water down here. So that means the water down here, which is normally relatively cold, suddenly starts to increase in temperature. Now, this is important because on the seafloor, there are things which we refer to as gas hydrates. They're a mixture of methane and water combined. And gas hydrates are stable. They can sit on the seafloor perfectly happily if the temperature is low and the pressure is high. Now, the water pressure doesn't really change very much. What does change, though, is the temperature. So if the water temperature right down there on the seafloor starts to increase, well, all of a sudden, these gas hydrates are going to become unstable. And if they become unstable, they're going to break down, they're going to release methane, and methane is 20 times stronger than carbon dioxide as a greenhouse gas. So a significant release of methane could lead to a runaway greenhouse effect. So that would then lead on to stratified global ocean, leading to oceanic anoxic event. Another possibility is that during this time period we have the eruption of flood basalts in Siberia. They're called the Siberian Traps, and it was an absolutely massive volcanic eruption. So it covers an area of approximately 200 million kilometers. So it's absolutely huge. So just think of the amount of magma that would have to have been erupted. And, this area is covered in, lava, layers of basalt. In some places they're hundreds, thousands of meters thick. A couple of kilometers of basalt in some places. So that's a lot of magma that's being erupted. So obviously that eruption had to take place over a very, very long period of time. So when a volcano erupts, what does it do? Well, one of the gases it puts out in quite large quantities when it erupts is carbon dioxide. And so obviously large quantities of carbon dioxide going into the atmosphere due to this, very, very large volcanic eruption is going to lead to global warming, which of course leads to an oceanic anoxic event due to stratified global oceans. So, you can see once again, we end up at the same place. So another thing that large volcanic eruptions will do is obviously they will put carbon dioxide into the atmosphere. Well, the carbon dioxide on the whole won't do that. Well, it will increase global temperature due to global warming, but some of that carbon dioxide will dissolve into the water. As that carbon dioxide dissolves into the water, it forms carbonic acid, and if you form enough carbonic acid, you are going to cause ocean pH to decrease. You're going to make the ocean more acidic. And as we've discussed, that's very, very bad for life in the oceans, especially if you have a shell made of calcium carbonate. There's also more though. When a volcano erupts, it will also put out small quantities of chlorine and fluorine. So chlorine and fluorine are halogens. They're very, very reactive. Now, if you put that chlorine and fluorine into the upper atmosphere, it will react with the ozone. So the chlorine and fluorine will react with ozone in the upper atmosphere. So slowly over time, if you have a big enough eruption going on for a long enough time period, the quantities of chlorine and fluorine going up into the upper atmosphere will slowly begin to degrade the ozone layer, which of course leads to increased quantities of ultraviolet radiation making it to the surface of the Earth. Obviously, volcanoes will also put out large quantities of sulfide gases. So sulfur-based gases, they will mix with water in the atmosphere. Once again, that will lead to the formation of acid rain, which can cause changes in ocean pH and also lead to the killing off of root systems of plants if that acid rain falls onto the surface of the Earth. So you can see once again, this large-scale volcanic event could have had very, very serious implications. But wait, it gets worse. So it just so happens that the area where the Siberian Traps was forming is also an area that has extensive coal beds. And so, well, what's going to happen when you put a hot body of magma in contact with coal beds? Well, one of a couple of things. Number one, you could actually set fire to those coal beds. Well, as you know, when you burn coal, you produce large quantities of carbon dioxide, which brings us back to global warming. Another thing that could happen is by heating up the coal beds, you could cause them essentially to begin to mature. And as they begin to mature, well, they're going to start breaking down. And as the part of the process of breaking down, they are going to form methane, which is going to lead us back to global warming again, which then leads us back onto a stratified global ocean and then eventually an oceanic anoxic event. So you can see, whichever one of these factors, gas hydrates, volcanism, or coal beds, leads to the formation of a greenhouse effect. Essentially, once you have a runaway greenhouse effect, it can then lead back to a stratified global ocean, which then leads to a anoxic event. So you can pretty much see what's happening. And then the final thing that's would have caused some stresses is the fact that the continents became more arid in the Permian. So when you start forming large continental land masses like Pangaea, well, that means it's more and more difficult for moisture-rich air to get into the center of large continental land masses. So if you think of Asia, if you ever look at a satellite image of Asia, what you'll notice is when you look at the center of it, you will see you have the Tibetan Plateau, you have Western China, and you have the Gobi Desert. That's a very, very dry area. You can also, argue that areas like, Turkmenistan, that areas around there are also, quite dry, semi-arid areas. And that's purely because those regions are so far away from the coast, so they don't receive large quantities of moisture-rich air. And exactly the same thing would have happened with the formation of Pangaea. You would have formed this huge supercontinent. The coastal areas would have been quite wet. The center of that supercontinent would have been quite dry. And so that would obviously mean that if any organisms that lived in the center of Pangaea that had been used to a relatively humid climate, all of a sudden would find themselves in a desert or semi-desert climate, and so once again that would have caused significant stress, and it would have led to the death of certain species. So you can quite clearly see that there are lots and lots of factors that could have individually cause the Permo-Trias mass extinction, or in conjunction with other factors could have caused the Permo-Trias mass extinction. So here's a very basic model here. So let's just say, okay, it was volcanism. So volcanism produces carbon dioxide, chlorine, fluorine, and hydrogen sulfide. Also, the heat from the magma begins to cause the coal beds to either burn or mature, causing the release of carbon dioxide and methane. That leads to global warming. That leads to reduced ocean mixing, that leads to a anoxic event, that leads to sulfate-reducing bacteria, that leads to acid rain, which decreases the ocean pH or maybe poisons land plants, that leads to a mass extinction, that decreases the uptake of carbon-12 by plants, causing this carbon-13, carbon-12 ratio to change, and of course that leads to an increase in the amount of fungi because they're feeding on all that dead organic material. So, as you can quite clearly see, the Permo-Trias mass extinction appears to have been a perfect storm almost. So we need to remember that just because the extinction was rapid, it doesn't mean that the event that caused it must also be rapid. So essentially what I'm saying here is that it could have taken a very, very long time for all of these factors to have built up to the point where we reach the point of no return. So once again, just like the roller coaster, you're going up and up and up and up and up. The factors are getting, building up. Then you reach essentially the crest. At which point, once you go beyond it, you're at the point of no return, and the ecosystem just collapses. It can no longer, support all these stresses that are causing it to, that are putting it under, lots and lots of stress. And so all of a sudden, the ecosystem just falls apart. And when that happens, you have a mass extinction. Now, in terms of explaining, why the mass extinction took so long to recover, that one's, pretty difficult to explain. If you had, a large-scale anoxic event, really, really affected ocean pH, that would take a very, very long time to sort out. So that might explain it to some degree. However, why land organisms took nearly 30 million years to fully recover, that one's a little bit more difficult to explain, and I can't honestly tell you that I have a good explanation for that. So, as you can see, the best model is you have a process that's building up to the extinction. It could have been warming up for millions of years before the correct conditions were actually achieved. At which point we are beyond the point of no return. Okay, everybody, so that's it for the first half of the presentation. Sorry it was a little bit longer than I would have liked. So remember, the code word for this presentation is coffee. I repeat, coffee. So please make sure you do the code word quiz. So as you can quite clearly see, the next part of this presentation is Paleozoic vertebrates and plants, but you're going to have to hold on to see that presentation for a little while. So take care, everybody, and that's me signing out.